Hello, everyone. This is Pocket Size Bio with Dr. Casco, and this video continues from the first part of Chapter 14, um, Pathogen Management Strategies. We're continuing on, and we discussed herd immunity, immunization with vaccination, right? And just in general, the idea of managing the spread of disease and the spread of pathogens, right? We're going to continue on section 14.2, right? And we're going to talk about disruption of a pathogen life cycle as a method to prevent and control the spread, right, of disease. So let's take a look. Oh, wait, let me take this down so you guys can see. Also, video here. All right. Okay. So, yeah, disruption of a pathogen life cycle, right? So, in order to disrupt the life cycle of a certain pathogen, of a certain parasite or um, um, protozoan or fungi or bacteria and whatnot, it's important to even know what the life cycle is about, right? And how it works and what are the stages, right? So, by understanding the life cycle of a pathogen, it can help, right, scientists figure out what are some critical points to inhibit the life cycle so you can prevent and control the spread of disease, right? So very, very important to know the life cycle of the pathogen and even so the life cycle of the vector, right? That you can kind of stop or prevent them from multiplying, which will enable you to prevent the pathogen from multiplying, right, and reproducing and so forth, right? So understanding the pathogen, right, life cycle in general, it aids, right, to prevent disease from spreading, right? And when we talk about the life cycle of a pathogen, right, it's very similar to what we talked about already, especially with a mosquito, right? You tend to have the reservoir or reservoir host, right? You need a portal of exit and a portal of entry and that whole mode of transmission, right? The actual replication of the pathogen in certain tissues of the host, of a particular host, and the susceptible host to continue the life cycle and essentially propagate the population, right? So um there's several ways you can target, the life of something, of a pathogen, depending on, the group of life that they're in and so forth. But um you want to be able to find factors that are, targetable, if I can say, right? That you can disrupt one one of these things, if you're able to stop the cycle at one of these places, you can prevent, disease transmission, right? And like I mentioned before, it could be any of these things, right? Now, of course, the method of replication and reproduction does vary, between a protozoan and fungi and a virus and a bacteria. So, depending on what the pathogen you're interested in, you need to figure out how to, what type of methods you can use, right? So, as viruses use host cells to replicate inside, you know, and basically use the cell's machinery and um, to compile all the structures to assemble the virus, and then the virus basically will be released out of the cell, right? And the cycle starts again and again. It infects a new cell, and then the viral DNA gets expressed, and so forth and so forth, right? Bacteria use binary fission, right? That we just talked about the previous videos. And, fungi and protists, they could use, sexual and asexual methods of reproduction, um, development of the spore or the sporangia, right? To propagate the species there, right? And so it's very important to understand the life cycle of the pathogen and if you want to stop it, right? So a word related to this that we're going to take a look at more in detail as we look at other diagrams is persistence, right? Persistence is basically how well, this um, um, this pathogen can, thrive in the reservoirs, or the ability for it to thrive even outside the reservoirs, right? Is persistence, right? It's a very persistent pathogen, right? It can thrive for a couple of days, a certain stage, right? Um, of the pathogen, right? A certain viral particle or bacteria outside of the host, it can still grow and propagate, right? So you have this word persistence, okay? And then when basically you're killing all the whole population, you basically are stopping the disease from spreading. You're killing the pathogen population, and when you reach a point that basically these organisms, these particles, if I could say, um, they can't transmit anymore, right? They can't infect, they can't replicate, they can't thrive anymore in the organism, and so forth. So you basically enable the disease to stop spreading, and at that point, hopefully, you eradicate the disease, right? Because the population isn't high enough to infect people anymore to cause them disease, right? Like I mentioned before, disease, then this is chapter 13, does require three interrelated factors, right? Mode of transmission, the susceptible host is in high enough population. More specifically, I want to say density, right? And the pathogen also, its population is high enough. And why is it high enough? Because there's a favorable environment for it to live in, right? So, Plasmodium is a very nice example where, remember, this is causing malaria, where you have the mosquito making the definitive host, or you're having the zygotes, and you're having the formation of the early individual. You have human that's serving as an intermediate host, right? If we take a look at this little diagram here, right? What would be some control strategies for the life cycle of Plasmodium, right? So, uh, you have a host that is showing early signs of the disease, and you can immediately get treatment, right? There are some anti-malarial drugs, right, that could basically um, can kill, right, certain structures that you want to prevent Plasmodium um to thrive in, right? Like chloroquine and primaquine. These can destroy, sexual forms of Plasmodium, the agent, or kill asexual forms of development of Plasmodium that will prevent development within the mosquito, right? So you're kind of killing off a whole method of growth, right, for the Plasmodium. And then you have ways to prevent the mosquito from stopping the life cycle, right? You have the mosquito as a vector. If you prevent breeding, right? If you remove any stagnant water that is accumulating during heavy rains and so forth, you're basically um, killing off larvae, right? You're destroying them for even growing in the water. If you prevent entry, some physical things in your house, that you prevent entry by closing the doors and windows, or prevent bites, getting mosquito bites in the first place, right? By using nets or sprays, right? You're going to prevent them from, having a blood meal, spitting saliva, and so forth and so forth. Though that in itself will prevent. Remember, what is the stage of the Plasmodium that gets into the body is the sporozoid, right? From entering the liver, right? So that kind of blocks off this stage as a vector, which will block off the Plasmodium so it can't continue on to the different forms, right? So there's several methods that you could stop a pathogen from its life cycle to continue, right? So, right, some control strategies, right? Okay, of course, exactly what I was just talking about right now. Mosquitoes, they transmit plasmodium between they ping pong between the human and the mosquito, the liver and the red blood cells to the gut of the mosquito to the saliva, salivary glands, and so forth, right? So, um, it's some other strategies is basically killing mosquitoes, insecticides. The problem with that is that you could get some resistance to insecticides. Adult mosquitoes have been resistant to some extent insecticides, so larvae are better fit um regulate mosquito populations. And the way that they do that is um they have poisons for larvae that they put in the water. That they put this powder in the water. It's a bit more common in developing countries, right? Which seems to work, right? Um, Plasmodium also, this is a problem that has shown some resistance to anti-malarial drugs, right? So you do need some um, kind of innovation in terms of medicine, right? Or anti-protozoan medicine. Okay. Um, right. So you're able to control targets, but again, there are some limitations, if I can say, or issues, that are faced. So um, if you target a human host through early diagnosis, right? How can you identify early diagnosis? It's a little bit difficult to treat, but the problem happens that you need to go to the doctor or you need to receive medical care. But even though you could get some early diagnosis, there are a lot of places that don't have medical care, and or the individuals can't really pay for consultation with a doctor and so forth and so forth. So, you know, it is hindered by poverty, access, access to health services, education, right? Education in the sense of understanding how pathogen propagation works, right? So what can you do? There are some other methods, like I was mentioning you about the powder, but that you just spread out. The government spreads and um puts powders in pools of water, right? That would essentially kill the asexual and sexual variants of the parasite, but drug resistance occurs, right? So that is a problem, right? Again, for the parasite and even for the mosquito, right? Where they can get resistance, right? So um this these two factors, can while it works to some degree, if overused, you can have more and more resistance. Okay. So um what are some other ways though that you can prevent this? You can have physical barriers, right? You can have mosquito nets or bed nets, right? Clothing to not even let mosquitoes, penetrate to the clothing and basically don't draw blood from you, right? Have these the little net filters on the windows, right? Using repellents all the time would reduce transmission, right? So again, but this is also a problem. A lot of people don't even want to put these nets or use these physical barriers. Just lack of diligence, lack of education, lack of awareness, right? Does unfortunately create this propagation, right? The spread of disease, right? So if humans can get more protected and avoid infection, then the disease would die out. How else would it propagate? Right? If you're not using one of the hosts, right? So it is a problem, right? Awareness is a problem. It's important to raise awareness, right? Um, so if humans can get more protected and avoid infection, right, the disease would die out. Travelers going to areas endemic to malaria should have preventive treatment, right, to avoid spread. Okay. So if you see this little figure here, this is a map showing predictive changes of distribution, right? Of Plasmodium falciparum, the one that for malaria, based on modeling data. Current distribution, you have it's quite interesting how you have everything near the equator, right? Not incredibly to the equator, but you have these areas in Africa, you have these areas in South America and Asia, right? But extended distribution by 2050 just because a climate has become more and more suitable, right? Okay. So, and then the ones in gray are basically current distribution, but they're unsuitable, would be thought of as being unsuitable by 2050, right? But that's not as important as the extension where you're seeing expansion of mosquito distribution, right? Which is a problem, right? So let me just go up here. Okay. So by what is another way that you could, try to mitigate this spread, right? There's many species of Plasmodium, right? And in this case, you have Plasmodium falciparum. But if you can identify a particular species, right, and you can sequence specifically certain proteins of a particular species, and you understand that it has this particular protein, you can create a more specific vaccine for a particular pathogen, right? So you can have higher levels of antibodies that are targeted, right? That target this particular disease that could um, come in and attack sporozoites from entering the liver cells. And if you have found a way to tag them, you basically can, destroy these cells more effectively, right? Effectively. So definitely bioinformatics and this whole idea of DNA sequencing helps in mitigating, right? And controlling and preventing the disease from spreading, right? So, but in general, you want to try to disrupt the life cycle of a particular parasite, of a particular disease or pathogen. Okay. All right. So we're going to move to another way, another control strategy, um, management strategy of spreading, right? Via medications, right? So this is one that we're all familiar with, right? At one point of our life, we've gotten some form of infection that we need to use antibiotics to get feel better, right? And some of the basic types of medication, and I'm gonna um talk about bacteria and fungi, right? You have streptomycin, right? And the streptomycin is a bacteria that can disrupt protein synthesis for bacteria. Penicillin, famous um, antibacterial medicine, right? It basically disrupts the cell wall, right, in the bacteria. You have clotrimazole, right? And this would disrupt cell membrane permeability, right? So depending on the type of antibiotic, you would destroy different things or inhibit different things of a particular organism, right? So medications, right? It's basically any type of chemical compound that can disrupt some aspect of its normal functioning, right? Um, and usually these medications against infectious diseases, they're referred to as antimicrobial, right? Agents that what causes the disease, right? But in general, you have these terminologies, right? You have antibiotics, which combat bacterial infections, antivirals, which target viruses and their life um, cycle, antifungals or antimycotics. They basically disrupt fungal life stages, right? And protozoal drugs like antimalarials, they can treat protist infections or persistent infections, right? It all depends on what the organism is, right? The target pathogen and the type of medicine, right? You can address, or you can focus on several things, right? Ribosomes. You can disrupt or break down ribosomes. You can disrupt DNA, specific sequence. Cell wall. Cell wall is very specific in terms of bacteria, right? Bacteria have a peptidoglycan cell wall. Fungi have chitin. Plants have cellulose. So they're very specific. Cell membrane, right? Or even certain pathways, right? Specific pathways for proteins, or creating other compounds like folic acid, um, which is important for the homeostasis of these particular organisms, right? So, in terms of antibiotics, right? I won't go into so much detail here, but it's you do need to know this, right? And I've mentioned this before, right? We did a whole experiment on this. Is basically these are medicines that inhibit or destroy bacteria only, and the reason for that is because of the method of inhibition, right? So what are they used for? They're used to treat an infectious disease. You can destroy the bacteria until you're cured, right? And as you basically break down the and lower the population of bacteria, you get to the point that you decrease the spread of disease to any from the infected host to any susceptible host, right? As we know, of course, bacteria are prokaryotes, right? So this is very easy to target because the physiology or the biology of prokaryotes against eukaryotes is quite different, right? So you can make special antibiotics, which destroys bacteria, but it doesn't hurt the eukaryotic cell, our cells, right? Human cells. So, in that sense, bacteria are prokaryotes, so it's easy to find, right? It's easy to target certain things because we're so different. Sorry. And develop drugs with side effects to humans, right? So, one of the famous groups of drugs, you can categorize them as gram-negative and gram-positive medicine, right? Because of their different composition of the cell wall that we talked about, right? So, yeah, if we didn't have antibiotics, a simple wound or surgery could become life-threatening, right? And it used to be like that before the advent of medicine, right? So, very famous is pneumonia, right? Or bacterial pneumonia, that a respiratory infection, bacterial infection that could cause pneumonia would return to being a mass killer, right? In your lungs, too much of a bacterial infection, you can't breathe, everything becomes very viscous and so forth, right? So, antibiotics can be classified as penicillins or cephalosporins, depending on what you're focusing at, right? But in these two cases, they interfere with the development or the synthesis of the peptidoglycan layer, right, of the cell wall. Okay. Then you have a second class of drugs. Okay, tetracycline, chloramphenicol, right, erythromycin, very, very potent antibiotics, right? So these guys, they don't address the cell walls, but they bind to prokaryotic ribosomes. So what happens there is that if you don't have the ribosomes functioning well, then you cannot synthesize proteins, right? That you need to survive, right? So, which is pretty good, which is quite interesting because bacterial proteins are much different than ours. So, there's not a problem if we have this type of medicine, right? Okay, so prokaryotic ribosomes are very different. So the ribosome itself, you can inhibit, right? Or you can break down. So it's quite different from eukaryotic ribosomes, right? So the structure is a little bit different. This is referred to as 50S strain of RNA, right? A ribosome is made up of RNA and some protein, right? It's referred to as 50-30 for prokaryotes and 60-40 for eukaryotes, right? The composition of between both of them, you don't really need to know, but just know that they're different, right? So because they're different, you can use a particular medicine, right? Like rifampicin, right? Piscin, ampicin. I need to double check if this is written right. Sorry about that. So it's used for treating tuberculosis, and that essentially inhibits, right, prokaryotic RNA synthesis. Okay. Again, guys, I'm not going over this again. Basically, antibiotics have been overused and misused, and why is that a problem? You're basically creating resistant bacteria, right? They rapidly evolve. Antibiotics in this case is the selective agent of natural selection and evolution of bacteria. So, and what is the consequence of that, right? Okay, very, very important. In terms of tuberculosis, right? They've basically become ineffective, right? That's how bad it's become. BT bacteria is important, right? If you guys remember that BT bacteria in the for um the cotton ball worm, right? Um, in any case, this is another type of bacteria, right, that has evolved resistance because infected people did not take a complete dose, right? So, and it does happen that a lot of people just they forget, are ignorant, they don't want to talk, they don't want to drink too much medicine. So it's does more bad than good when you don't want to skip a dose, right? Or when you skip and don't take your course. Okay. And it depends on the type of antibiotic, right? Like some antibiotics you need to be taking in course with other antibiotics for a good period of time to be completely effective, right? Especially when you're having surgery. Sometimes during surgeries, pre-surgery and post-surgery, you need to be taking some antibiotics, the full course, right? Because let's say you did a surgery that you need to remove an organ, right? Um, or you have an infection, you need to take all these antibiotics to, ensure that that pathogenic bacteria doesn't overgrow. All right. Okay. Antivirals is very similar situation, but in terms of antivirals, right? You try to, focus on a chemical to inhibit, the ability for viruses to replicate. Pretty simple, right? Only works on viruses. That's why they're referred to as a viral antivirals, right? And of course, if you reduce the life cycle of the virus and if you inhibit it, there's less viruses, so the duration of the disease will decrease, right? So, um, antivirals, they treat viral infections by minimizing the symptoms and infectivity, right? Um, Generally, antivirals help also mitigate the symptoms. Um, Doctors rely more on the natural immune system rather than antivirals. Okay. Um, but yes, there are some antiviral medications, right? So most illnesses, and this is quite interesting, that most illnesses due to viruses are treated symptomatically until the host immune system controls and eliminates the pathogen, right? So you rather treat the symptoms rather than try to eradicate the virus from within the body. Basically, the host, our immune system would take care of that. We just kind of ease the symptoms, right? But there are some antiviral drugs that can um, a, target, if I can say, specific enzymes, right? Moreover, enzymes involved in viral nucleic acid synthesis, right? What are some examples of these? You have HIV, herpes viruses, which are grouped into the cold sores and the genital herpes, right? Herpes themselves is a class of viruses, right? Hepatitis viruses, they basically cause liver cancer, right? And related diseases to liver, and influenza viruses, right? I'm gonna get to this little diagram in a bit, right? So it's important to understand that creating safe and effective antiviral drugs is a little difficult, right? It's a little difficult because one, true, the virus changes, but also the viruses use the host cells to replicate. That means if you want to stop the virus, you need to create some antivirals that only address the virus, right? But the problem is that the virus uses our machinery. So you'll be trying to attack our own cells, right? So this makes it difficult to find targets for the drug that will interfere with the virus without harming the host organism cells, right? So that's a big problem, right? Another problem is that, DNA strands for viruses or RNA strands for viruses, they tend to um, uh, you tend to get new strains, right? New DNA strands because they change relatively quickly, right? And if they change, then the protein changes a little bit, and then the vaccine has to change, right? That they give us, or else, our natural immune system won't work as effective, right? So there's several points that you can target, a virus using antivirals, right? You can, and I'm just going to zoom out a little bit, right? So these antiviral chemicals or drugs, right? They can inhibit the virus from entering a particular cell. Okay. Usually these envelopes, right? This protein coat has little receptors that bind to the cell membrane, which initiates endocytosis, right? Enables it to come in, right? So this is referred to as entry blocking, inhibiting binding or attachment, entry blocking, right? You can inhibit the entry or penetration, right? You block protein channels or receptors, right, in the whole cell membrane. If that doesn't work, you can inhibit transcription, right? Remember, the DNA goes into the cell and then into the nucleus and attaches itself to the host DNA. But maybe you can prevent the DNA from getting expressed, right? Or turned on, right? So inhibiting transcription could work also, right? So let's say the DNA managed to get inside the host genome, right? Okay, that's fine. But maybe you can block the transcription of the viral DNA of the viral DNA, so it doesn't produce, all these the components of the virus to make more viruses, right? So by inhibiting transcription, you could inhibit new populations of viruses forming. Okay, so um, and then you can also focus at the portal of exit, where basically, let's say transmission did pass, and then you're getting translation. The protein gets assembled, and then you want to ship it out. The virus basically gets complete and wants to be released. That portal of exit could also be a place to target. Okay. So to prevent the release of newly assembled viruses, right, from the cell membrane and outwards. Okay. So these have been used to treat a wide range of influenza strains, right? Influenza generally caused by two types, influenza A, influenza B. Every year, there are one of two types of each influenza A and B that is circulating around, right? And again, influenza could be treated for antivirals, right? Oseltamivir. Oseltamivir is a very famous type of medicine that you could use to treat antivirals, right? But there's something even better, which is prevention, right? Vaccination. If you can get a vaccine for these newly, change viruses, then maybe you can also help prevent the spread, right? It is interesting, this last thing. I remember putting it here because um, no one really knows how um, there's no significant evidence, right, of resistance to antivirals in circulating strains of influenza, right? So far, so good. Take your full course, take your vaccinations. Everything seems to be working for now, right? Hopefully, it always stays like that, right? And so forth. All right. Good. So we get into, oops, yes, yeah. What are some other ways, strategies to help control the spread of pathogens? You also have, sorry, guys. Okay. Physical preventive measures, guys. Very, very important. This is something we can all practice. We should all be practicing, right? Um, Physical preventive measures, right? So, number one, hand washing. Always wash your hands, guys, after you eat, after touching, any surface, or periodically. It's just good to wash your hands, right? One of the more useful measures to spread against the spread of disease, right? Because if you have your hands dirty, you put soap. Soap has these you know, bacterial properties that they can destroy the cell wall, and they can also just you know clean everything out, just slip it out from your hands, right? So very important, right? Regular hand washing can prevent individuals from contracting infections, especially those that are spread by fecal, oral, or direct contact routes. Okay. Keep your hands out of your mouth and wash your mouth and wash your hands often, right? On a global scale, hand washing can significantly reduce the mortality from certain infections, especially diarrheal diseases or illnesses, right? Very, very important, right? Most effective if warm water plus soap is used, right? You guys have probably heard antiseptic hand wash, right? That you can destroy this bacteria, right? And again, in general, they have a antimicrobial substance which basically um can either rupture the bacterial wall, right? Or it could inhibit their growth, right? So um this does help stop the spread, right? Hand and soap washing, right? You can also use alcohol. 60 to 95% alcohol can destroy the cell wall of bacterial cells and viruses, right? More specifically, the viral envelope, right? So, you know, washing your hands, rubbing on both sides of the hands and between fingers, guys. I'm sure we all remember washing your hands during COVID and how to wash your hands correctly, right? Um, thoroughly rinse and dry, right? Um, when should you do this, right? Of course, you're not. Also, you don't need to be every 5 minutes. It doesn't even matter if you haven't touched anything, right? But there's times that you're more susceptible uh before preparing food or eating the food, right? After sneezing, coughing, gardening, remember the soil-borne bacteria, right? Toilet duties, looking after sick people. All these are scenarios where you should definitely, if you can, wear gloves. And and even after wearing gloves, throw away the gloves and then wash your hands, right? Very, very important. Okay. Under certain circumstances, right? Especially being exposed to an infected host of a disease or the spread via contact or fomites, right? Wash your clothes. Take a shower, washing of clothes. How many of you, take a shower after a long day? You're exposed to all these things, right? All of our skin, our clothing. It's always good, just put your dirty clothes in the laundry, go take a shower, just clean yourself from the day, right? Uh, um, Reduce the chances of bringing a sickness into your home, right? It's true. It's a good practice, right? I'm pretty sure we all do that. Definitely recommend that, right? Washing your clothes, right? So washing removes or kills the pathogens, right? And prevents spread of disease. Okay. Very, very important. So disease spread by fecal or oral or direct contact routes, again, avoided through hand washing. Um, that shouldn't be a big problem. Just after you use the restrooms, just clean yourself well all over the place and um, wash your hands well, right? Um, and so forth. Okay. Um, Anytime you're exposed, right, with an infectious agent, right? Maybe your hands are dirty and then you're cooking food, easily you can contaminate the food. Easily, easily, easily. All right. So, um, and also if the food isn't cooked well, right? So you want to cook well, have some clean hands, cook, and so forth and so forth. This is even if you help your parents cook food, right? So, yes, uh, or else you're gonna basically you're inviting all the whenever you eat, you're inviting every all the bacteria, all the pathogens to get inside your body from the digestive system, right? Your gastrointestinal tract, gastro. Okay. Use filtered clean water. Use filtered clean water and food, right? Right. Water should be treated before being supplied. Definitely use filters in your house, or just buy bottled water. I buy bottled water. It's definitely not good for the environment. um, uh, Clean water, right? If you don't have a tap, if you don't have bottled water, just boil your water. Boil your water, right? Salad ingredients, right, are washed in untreated water. You can easily get infectious agents on the salad, and then you just eat it. You're basically welcome, welcome, come, doors are open, come into my body, right? So you want to clean thoroughly. I've heard some people put like drops of chlorine on salads to help clean the salad, and then they rinse the salad, right? Um, yeah. All right. So pathogens, they could be foodborne, right? We've talked about different forms of borne: waterborne, soil-borne, airborne, right? Foodborne pathogens, right? Because basically the food that they occupy is undercooked or out of date. You never want to eat expired food. That's just the reason why there's an expiration date on food labels, right? Is because there's a kind of statistical analysis of when you're likely to have a population of bacteria growing in that food already by that time, right? Just avoid, right? Date of expiry or expiration dates. Be careful. Just throw it away. Don't even open. Don't even try. Even if it's sealed, it's in the fridge. Just avoid getting sick, right? Avoid getting sick. In general, sanitation is another factor there, right? Safe disposal of human poo or urine. When we use the bathroom, we're just liberating a lot of bacteria, a lot of um, potentially pathogenic bacteria that's coming from our bodies, but they're not in high populations, right? Unfortunately, 2.6 billion people in the world lack adequate sanitation. A lot of people drink water that is contaminated. And it's 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 ironic that the water they drink will give them diarrhea, which will further make them lose water, an infection, and then loss of electrolytes, aside from the whole pathogen getting in there, the disease infection getting inside your body, right? Many governments build sanitation infrastructure for the communities, but they do so, if they have the funding. Funding is so important, right? Underdeveloped countries need funding so they can build better sanitation measures for all these poor communities, right? Unfortunately, this whole idea of excrement, right? The feces and black water, contaminated water, especially in urban areas, they have to be um, efficiently removed, right? Basically, the whole system, our whole drainage sewage system, some places very effective, some places absolutely non-existent, right? So um, it gets combined with water, river water, and so forth and so forth, that easily you can contaminate anybody, right? So the measures to prevent that oral entry of many infectious diseases is important, right? Because it can slow the spread significantly. Okay. All right. And lastly, I'll end this video here at this page. Physical preventive measures, right? So I kind of talked about this before, but you know, um, behaviors, right? Just good etiquette, right? When you want to sneeze or cough, cough into the elbow. Don't just blah into the air, especially if you're in a confined area, right? Cover your coughs, cover your sneezes, right? Sometimes you have to actively be aware, but then it becomes a reflex. Um, you know, yeah, the elbow can act as a barrier to airborne droplets. It stays on your shirt rather than spreading to other people, right? Um, as we know, as we're very familiar with COVID, and tuberculosis can easily spread to coughs and sneezes. The elbow will capture the pathogen, right? If the hand is used instead of the elbow, you'll basically use your hand as a fomite. So you also need to wash your hands. But it's better, if you're using your hands, it's better than letting it spread out into the air, right? Um, yeah, and then you could just wash your hands. Other barriers, depending on the type of pathogen, like mosquitoes, I mentioned before, right? Barriers, very, very important barriers, right? You have mosquito nets. Huh? I would actually sleep in a net like this back home. Would feel like a little princess. Ridiculous. But it works. It absolutely works, right? Mosquito nets spread greatly inhibits, right? Almost no mosquitoes passing in. Kitchen gloves when you're working with when you're trying to clean up, the kitchen area, or you see some mold there. Um, mm, um, Surgical gloves, many people use that nitrile gloves to cook. Very nice. Doesn't contaminate your hands with certain bacteria. Surgical masks for airborne diseases, right? We all use masks for influenza to stop the little air droplets. Very, very important, right? Um, um, and just in general, the personal protective equipment, if you guys remember from COVID, right? I'm going to talk about this in class because it's the type of mask that people would custom make. It, it was inspiring. We'll see. We're taking a look at some pictures. Anyways, mosquito nets have been more effective when sprayed with insecticide, definitely, right? So you have the physical barrier and you have a chemical protection there, right? Um, these diseases of malaria can last for months, right? Susceptible hosts need to be diligent, right? So especially if you're living in these areas, right? You need to be able to prevent yourself from getting infected, right? Tuberculosis, Mycobacterium tuberculosis, you want to avoid that spread of coughing and sneezing, wear masks, right? Because why? The mask stops the inhaled droplets. Of course, the virus or bacteria may pass through the mask, but they're in the droplet. So if you stop the droplet, they're not gonna, wiggle into their mouth. It does have some protection. I do recommend changing your mask throughout the day, right? And so forth. But if you can't, then what can you do? Just one mask is better than nothing, right? Um, Definitely, and you see healthcare workers always wearing a mask. You want to be protected from the nuclei, right? From the droplets containing the pathogen and so forth, right? So very, very important during COVID. Okay. Hand washing, incredibly important, guys. Throughout the day, wash your hands all the time, all the time. Okay. You're going to kill the bacteria. All right. So, we're going to take a look at this in class, but this is imperative that you study, right? For the ATAR, for the unit test. Very nicely laid out management strategies, right? There's some questions on management strategies in terms of the different um, diseases. You should know some specific examples. We're going to go over this, right? On how to, check prevention, early diagnosis, and treatment, okay, of all these diseases: influenza, Ross River, honeybee, ABL, right? Tuberculosis, tetanus, and so forth and so forth. These tables, guys, please study for the ATAR. Please, please study for the ATAR. All right. If you can't even study everything that we talked about, maybe this is a good summary table, even, right, for you to take a look at. Okay. So we're almost done with chapter 14. This basically deals with COVID, and then we get into um, um, monitoring, right? We're almost done with the last chapter, guys. Okay. The last thing we'll talk about is these 4 little sub panels, which is monitoring disease activity. So here's where the epidemiologist comes in. Okay. But that'll be another video. That'll be part 3. All right, guys. So thank you for watching and take care.